Hello, students. This is Dr. Faizan Mirza. I'll be covering questions related to immunity in this particular video. So this is students. This question came in May in 2019, May in 21. The question asked, they have provided to us the molecule of antibody, and they are showing there's a ribbon model of the antibody molecule. They are saying, describe how figure 1.1 shows their secondary and tertiary structure of the antibody molecules can be seen in the figure. So if you refer to the figure here, figure 1.1, you can see the beta sheets. So these are the beta sheets. You can see they are turning, so they are taking turns, and all these strands, this actually are beta sheets. So these are all beta sheets here. So you can see beta sheets at various places within this antibody molecule. So this basically shows the secondary coiling. Plus, you can see these coils as well. Now, the coils that you see here, they are not exactly the alpha coils which we see here. All the alpha coils can be seen at these points. So these are some random coils. So a very short alpha coil and then some random loops, and these are all random loops here. So what can we deduce about this, that this is actually a secondary structure? So what we'll be stating, we'll state that the antibody molecule can be seen to be having random loops as well as beta sheets in all the polypeptides. How is it a tertiary structure? Because the antibody molecule can be seen to be folded in a 3D shape, because we know that tertiary structure is all about the 3D folding, and this is how the R group, because of the R group interactions. In the next part, they have shown a transmission electron micrograph. What's a transmission electron micrograph? Transmission electron micrograph basically allow electron beam to pass through the specimen, so you can actually see the internal structures of the specimen. So the internal structure of the specimen can be seen because the electron beam gets transmitted through the specimen. This is the hybridoma cell. They are saying hybridoma cell in figure 1.2 synthesizes and secrete antibody molecules, synthesizes and secretes molecules of monoclonal antibody. State the role of the structure. They build X and Y in production of the antibody molecule in a hybridoma cell. X and Y are actually able to use. So let's go to the diagram. What is Y? Y is actually a circular structure, and inside the circular structure, you can see these finger-like projections pointing inwards into the circle. So basically, this actually is mitochondria. The mitochondria is being looked around along its you can say widest portion, or you can say along its circular axis. If you just turn it around, the mitochondria would look lengthwise and it would appear the rod shaped structure. So this mitochondria is actually appearing circular because you are seeing it along its circular way. So this is a mitochondria. We know it will provide ATP for the synthesis and secretion of the antibody molecule. What's this structure? Since these are cisternae, you can very well see these are actually cisternae, and these cisternae could very well be the cisternae of ribosomes, rough ER, because you can see the ribosomes here. But at the same time, rough ER is actually not present always, mostly at the periphery of the cell. Periphery means the outer end of the cell. You can see that this is the nucleus of the cell. So rough ER should be somewhere here around the nucleus. This is quite away from the nucleus, although rough ER, if you label this as a rough ER and you mention that its function is to synthesize proteins and antibody molecules, they would allow that answer as well. But a more appropriate answer of this is actually Golgi. Why is it Golgi? This is Golgi apparatus because this is having cisternae, and we know Golgi is involved in polypeptide antibody, or you can say they folding it together, R group interactions, and getting modified and all. And that's how you can just work this out. The hybridoma cells produced for production of monoclonal antibodies involves a number of stages. One of these stages is formation of hybridoma cell. Outline the stage in which hybridoma cell are formed. So you don't really need to explain the entire monoclonal antibody production mechanism or procedure here. You basically need to just highlight the stage at which hybridoma cells are produced. So hybridoma cells, basically, they are produced when the plasma cells that have been screened. You screen plasma cells because when you take plasma, the spleen out of that mice, that spleen will be having a lot of plasma cells having the ability to secrete different kind of antibody each. So we basically need the plasma cell which is releasing our the antibody that we are interested in. So we'll be screening them, and these plasma cells will then be fused with cancer cell, the myeloma cells. There is the cell membranes will fuse together because the cell membranes are of phospholipids, and they will result in a cell called hybridoma cell. The fusion of these cell is triggered by passing electric current, because when you pass electric current, the phospholipids, they start to move about in a different manner. So the cells can fuse, or you can use a chemical called fusogen. So either of them can. Outline the use of monoclonal antibody in the treatment of disease. So monoclonal antibodies can be used in treatment of diseases because the antigen of the disease is used in the production of this monoclonal antibody. We know that the monoclonal antibody will have the variable region and the variable region at which the antigen binding site is. So that antigen binding site will be complementary to the antigen that the disease is actually having. So for example, trastuzumab, it is a medicine which is used in the treatment of breast cancer. These monoclonal antibodies have a variable region complementary to the antigen on the target site. Plus, we can also state that the monoclonal antibodies, when they are used in treatment, you have to go for multiple doses. So multiple doses are required for the treatment of a disease when MABs or monoclonal antibodies used as a mode of treatment. If they were asking you about diagnosis, then for diagnosis, a single dose is enough. You don't need to reapply that unless the diagnostic procedure need to be repeated, although it's very specific, so it's done just with a single dose only. This question here is from May, June 2015, variant 21. The question is about how the pathogens they enter in the gas exchange system, but then it goes into the immunity part of the topic. So initially they have shown to you the electron micrograph of a bronchiole. You can see this is ciliated epithelial lining. These are all epithelial cells having cilia. So these are all cilia bearing cells. And in between you will have a mucus producing cell or a goblet cell, and that goblet cell will be releasing mucus into the out of that bronchiole, which will spread out on the surface of the bronchiole. So we know that under the bronchiole, in this layer, there will be a blood vessel, because the blood vessel, you can see this lining, this line separates the blood vessel, and all these cells are the red blood cells. They don't really look biconcave because they are in the various, you can say, floating. They are floating all inside the plasma in blood, so they don't really look biconcave. But since they are all shaded off, they do not have any contrast inside, they don't have any organelles inside, so definitely they are red blood cells only. So they are saying in the tissue X and the cell Y, tissue X is epithelial or epithelial lining or epithelium, and cell Y is a red blood cell. With reference to structure visible in figure 2.1, states three ways in which the lining of the trachea, bronchus, and bronchiole provide protection against the entry of the bacterial pathogens. So we know that cilia, it filters and move the mucus carpet towards the throat, so we get a mark there. We are just highlighting what cilia is doing. Then what's the mucus doing? Mucus is released by goblet cell. It is released by the mucous glands as well. Although mucous glands are not shown here. If mucus gland was shown here, the mucus gland is not in this line. The mucus gland lies below the upper layer of the lining of the trachea, bronchi, and bronchiole. So mucous gland will never be present here. It will be present somewhere under these epithelial lining. Anyhow, so just recalling that. So here you can say that the germs they get adhered to the mucus here. What's another thing? What's another function of this mucus and epithelial lining? Together they provide a barrier, and the barrier prevent the pathogen from penetrating into the bronchiole. Since we are discussing immunity here, so this diagram is a very coherent diagram to understand how the immune system works. So this is, you can see an entire description is shown to you in figure 2.2, how the bacterial pathogen enter. So this is the bacterial pathogen shown to you. You can see the bacterial pathogen is having these triangular structures on its surface, and these triangular structures are the antigens which are there on the surface of the bacterium. This key is given to us. This is the secretion of cytokines. So wherever this is mentioned, that basically means that cytokines are being secreted. If cytokines are being secreted, which cells secrete cytokine? T helper cells, they secrete cytokine. So we can identify that, okay, this entire story is about T helper cell. We know T helper cell bind to antigen, and this antigen is being displayed by a cell that got infected. How the cell got infected? These are the bacteria. These bacteria gained entrance into the cell. They infected the cell. The cell ended up digesting out a few of these pathogens, and it displayed the antigens of that pathogen and the bacteria on its own membrane. Basically, this infected cell is behaving as an antigen presenting cell. Now, when the antigen presenting cell is there and the antigens are there, the antigens, they can interact with three major cells of your immune system, the three lymphocytes here, and the three types of lymphocytes here. In fact, two types, and the third is a subtype of a T lymphocyte. So we have cytotoxic lymphocyte. Cytotoxic lymphocyte is having a T cell receptor. This structure here is a T cell receptor. Similarly, this helper T cell, this is also a T cell receptor. They both will interact with the antigens on the surface of the membrane. So when they interact with the membrane, what will happen? The cytotoxic cell will basically just take its membrane close to it, and it will drill a toxic vesicle into the cell. When the toxic vesicle goes in, it will kill this entire cell altogether. That's the killer T cell, how it functions. Now, helper T cell. The helper T cell is also having a T cell receptor present on its membrane, complementary to the same antigen. it divides to form memory cells and cells that produce cytokines. So these are all memory cells. So this is, you can see, these are the memory cells of the helper T cell. And these are the cells that are releasing cytokines. We know the cytokines are required for conversion of the B lymphocyte. B lymphocyte, they differentiate to form plasma cell and memory cells. So cytokines are required there. You can see that this is the antibody receptor, which is present on the surface membrane of the B lymphocyte. It binds to the same antigen here. So in this case, the T helper, this T helper having the T cell receptor, the killer T cell having the receptor, and the antibody receptor on the B lymphocyte, they all three of them are complementary to the same antigen, which is present on the membrane of the antigen presenting cell, that in turn belongs to the bacteria. So the B lymphocyte here then divides to form plasma cell and memory cell. They divide by by the process of mitosis. We know that memory cells, they provide long-term immunity, the immunological memory. Plasma cells, they start to secrete antibodies at a fast rate. You can see antibody molecules are being secreted. Now they are seeing straight what's happening at the stage J. This is the stage J. Stage J is actually how the infected cell becomes an antigen-presenting cell. The bacterial pathogen is entering into the cell by endocytosis. The cell presents the antigen on its own membrane. Explain the role of cell L at the stage K. So cell L, we know the cell L is the infected cell that is behaving as an antigen presenting cell. What's the function at stage K? So cell as behaving the antigen presenting cell, it displays the antigens on the bacteria on its own membrane. T cell with the receptor complementary to this would get activated by, and clonal selection would occur, and they would start to just release cytokines. You need to explain how the response of the second infection of bacterial infection. We'll just explain how the secondary immunological response basically occurs. So since already there will be memory B cells circulating, they would recognize the pathogen upon the second invasion. The memory cells will then divide by mitosis to form more memory cells and plasma cells, and antibody will then be secreted by the plasma cell quickly, and this increases the rate of phagocytosis. Clonal selection, clonal expression is not required in the secondary immune response. Clonal selection and clonal expansion is never required. There are various ways in which the effectiveness of immune response can be reduced. How the following reduce the effectiveness of immune response? HIV, a number of T lymphocytes, they reduce in HIV. So how it reduces the effectiveness of immune response? The helper T cell count, it decreases because the HIV particles, they live inside the cell. So the helper T cell, they start to die. This is how it reduces the effectiveness of the immune system. Some pathogens are covered in the cell membrane from their host. So here you will be highlighting antigen concealment. For example, the malarial plasmodium, as well as TB, HIV, they all live inside the cells, and they hide from the cells so that they basically conceal their antigens from the bite itself. The cell surface marker would be inside the cell, would basically hide, and they would not initiate immune response. B lymphocytes do not mature properly and do not recognize antigens. Now, when they will not mature properly, where do they mature? They mature in bone marrow because they are B lymphocytes. So they would lack the proper antibody receptor molecules, and they would not identify the non-self antigen the way they should. That's it from my side. I hope that was helpful for you. Thank you so much.